Thank you so much again for inviting me to speak. So I'm at the University of Michigan. I'm in both the Department of Epidemiology in the School of Public Health and at the Institute for Social Research. And much of my work really focuses on integrating sort of social factors as well as genomic and epigenomic determinants of age-related diseases, including cardiovascular disease, dementia, and their risk factors. And so, as Ryan said, today I'm going to talk about social epigenomics, which I think is a really exciting area that's pertinent not just to chronic disease, but also to infectious disease. So we know that the social factors can be big influences of chronic disease risk. And that really includes things both at the individual level as well as the neighborhood level. So at the individual level, we think about things like social or socioeconomic factors, like someone's education, their income or wealth, occupation, or other things that make up what we think of as a socioeconomic status or socioeconomic position. And at the neighborhood level, we also have some of those things going on. So we have socioeconomic indicators, which could be, related to things like aggregate levels of income, education, wealth, and things like that. Racial segregation, and things that are more like perceptions, like safety. And these social factors can really increase risk for a lot of different chronic diseases, including things like cardiovascular disease and dementia, as well as their risk factors and other diseases like asthma and mental health disorders. And so we know that SES is a major determinant of, for example, cardiovascular and cognitive health. And that, for example, it's associated with all kinds of metrics around those. So increased incidence, increased prevalence, increased mortality. And we also know, importantly, that the socioeconomic factors and social factors operate sort of across the life course. So both adult and childhood socioeconomic and social risk factors can be associated with late onset or adult disease. And we also know that these relationships to disease manifest as what we think of as socioeconomic health disparities, but also to keep in mind that that also really overlaps with race ethnic health disparities, because in the US, it's unfortunate, but because of racial segregation and other things, the socioeconomics and race ethnic disparities are often intertwined. When we talk about neighborhoods, we're really talking about the places where people live and where they work. So these can be defined really at the census tract level or through other administrative boundaries, or by some kind of distance or radius to someone's residence, for example. And neighborhood level characteristics also influence these diseases, independent of individual level socioeconomic factors. So digging a little bit deeper here, when we're thinking about things like the socioeconomic environment, as I sort of said before, it's sort of these aggregate levels of individual SES, like average income and poverty. And also, even independent of that, the racial composition or the segregation in neighborhoods also can be an independent predictor of cardiovascular and cognitive health. There's also pieces around the physical environment. So can you walk in your neighborhood? Can you get access to healthy food and those sort of pieces? And then there's also the social environment. So we can think about things like, as I said, neighborhood safety. We can think about things like aesthetic quality. So does your neighborhood, is there trash on the streets? Is it like a place where it feels like a place where you could walk around? And how is the cohesion among neighbors? And so we found that, living in a disadvantaged neighborhood has also been linked to increased mortality and increased cardiovascular and dementia risk factors, especially things that are more cardiometabolic, like hypertension, obesity, and diabetes. So people who have a low SES or who are living in disadvantaged neighborhoods have some characteristic biological patterning that's more prevalent, including dysregulation of multiple types of hormones, including the cortisol, which is a diurnal stress-related hormone. It's part of the HPA, or the hypothalamic pituitary adrenal axis. There can also be dysregulation of more immediate stress hormones, so catecholamines, which are part of the sympathetic nervous system, are related more to immediate reactivity to stress. And both of those systems show sort of wear and tear after a while that causes this dysregulation, because chronic psychological distress can really wear on these systems. The other one is chronic inflammation and changes in immunity that can be linked back to some of these low SES and disadvantaged neighborhoods. And so we think these common biological pathways might influence multiple risk factors for these chronic diseases. And because we're epidemiologists, we all love DAGs. This may or may not be a DAG. I think it is. It's got a couple of bidirectional arrows. This is sort of the conceptual model of ways that the social factors might influence these chronic disease risk factors. So you've got your individual level factors, your neighborhood characteristics, and the stress and inflammatory pathways are two major pathways that we think are affected by these. And those do interact with things like health behaviors, which also may be mediating some of these relationships. And then we can't forget that in the neighborhoods in which we live, there's also environmental exposures or pollutants. And so those can also act as mediating pathways. And so much of the work that I do kind of looks at mostly the stress and inflammation pathways, but thinking about ways that the epigenome might fit into this diagram. And so if you're not familiar with epigenetics, I'll just remind you. So epigenetics is the study of mitotically heritable. So when cells divide, those epigenetic patterns are preserved and passed on to the next generation of cells, and potentially reversible changes in the regulation of genes that occur without a change in the sequence of nuclear DNA. So you all remember probably that your DNA is a double helix. It's made up of A, T, C, and G bases. The epigenetics is really like a layer that sort of sits on top of the sequence of DNA. And so when your epigenome changes, it doesn't change those ATCs or Gs, but it interacts to tell how those genes that the ATC Gs are coding, how are they expressed? When are they expressed? Where are they expressed? And how much? And so we call that the regulation of the expression of genes. And that's what the epigenome is really important for. And two of the major mechanisms of the epigenome are DNA methylation and histone modification. There are also others. So DNA methylation is the one that is most commonly studied. It's kind of the simplest one. It's the easiest one to measure. I'm going to talk about today exclusively DNA methylation. So in humans, DNA methylation occurs at a C or cytosine DNA base that's followed by a G. So this is a cytosine, this is a guanine. And at the cytosine followed by the guanine, you can have a methylation mark, or you can have, or it can be unmethylated. And so the methylation is really just a covalent modification to the C base by adding a methyl group or a CH3. So you can kind of see that down here. You've got your regular old cytosine base. And at this position here, there's a carbon right here in this ring, and a methyl group can get added, and that's methylation. And so DNA methylation is commonly, we measure them at DNA methylation sites, or what we call CPG dinucleotides. So that's just here where you've got the C followed by the G. You'll see me refer to these as CPG sites. So that's places, again, where we can have methylation or not. And DNA methylation really occurs throughout the genome at varying places. So places in the genome where a C is followed by a G are much less common than you would expect by chance alone. And when you find them together, they're usually in these things called CPG islands. So this is where you have a lot of methylation sites that are potential methylation sites that are clustered together. So here, I'm just trying to give you some of the terminology. So first I'm showing you here, here it is. First I'm showing you a gene. Imagine that this is a gene, and here is where it's coding for protein. And upstream of the gene, or there's a place that's sort of the gene promoter. So at the gene promoter, it is a place where some of the cellular machinery will sit down on the DNA and express the gene. And the promoter regions are really important for the regulation of gene expression. And CPG sites that are in promoter regions tend to be really strongly associated with the expression of those genes. In addition to things like the promoter site, these special CPG islands where you have a lot of CPGs clustered together, they also tend to be found a lot of times in the promoter regions of genes. And so we pay special attention to the promoter region, and we pay attention to these CPG islands because it's been found that not in the island itself, but just nearby the island in what they call the shores and shelves, the CPG sites there also tend to be very important for things like cell differentiation. And so we think they might also be important for regulating gene expression that could be critical for diseases. So here's where we sit now. So social epigenomics is really thinking about how the epigenome mediates the influences of social, psychosocial, and neighborhood factors, and even things like environmental toxicants on chronic disease or cardiovascular disease and dementia in my case. And so the first half of the talk is sort of here. I'm going to focus on this first red arrow, where we're looking at the effect of these social factors on the epigenome. And since the epigenome is likely to exert its effect on disease through modifying gene expression, I'll also look at these purple arrows. And so kind of looking at the pathway whereby the social influences affect the epigenome, and then the epigenome affects gene expression, and that eventually leads to disease. And so all of these red and purple arrows will be pieces in the talk. And I want to emphasize too that all of this is taking place within the context also of the genome. The genome is also important. So the genome can influence the epigenome itself. It can influence gene expression independently of the epigenome. And it could have also other effects on cardiovascular disease or interact with social factors. So just keep in mind, I'm not going to talk too much about genetics, but it is sort of there in the background. Okay, and I think I've convinced you of this, but just to sort of reiterate, and I'll just give you quick examples. This area of social epigenomics really is motivated by the fact that these social factors influence adult onset diseases, and that humans adapt biologically to changing environments, and methylation is modifiable, and it's modifiable by your environment, and it influences gene expression. And so where this sort of interest started was that animal studies had shown that DNA methylation or gene expression at some key genes like stress response genes and inflammation response were changing in in people or in animals at that point, and now we see in people who are having those kinds of social stressors. And that those patternings can actually last and persist across the life course. And so, one of the first animal models really kind of picked up this pathway, which is where they looked at in rodents, they had early life stressors, and it was characterized by these lifelong dysregulations of the HPA axis here. So you can see that with the HPA axis, you have the brain is releasing hormones that trigger the anterior pituitary to release adrenocorticotropic hormone, which causes the adrenal gland to release cortisol. And cortisol is how we measure a lot of times sort of stress in humans. And this is really the pathway that becomes dysregulated over time. Again, that we saw in animals, that's accompanied by changes in methylation and gene expression. Another line of evidence, this was just so interesting when this first came out, and still very interesting, obviously, is so Steve Cole at UCLA did a lot of work on social adversity. And he showed that a variety of adverse environments, including things like low SES, instability, loss, and trauma in humans, and other things like social isolation in primates and in rodents, lead to these really characteristic patterns of gene expression. So this is called the conserved transcriptional response to adversity. And in these earlier studies, they were looked at in leukocytes, which are white blood cells. And what he found essentially was that these adverse events are stimulating the transcription of genes that encode pro-inflammatory cytokines. So they're increasing inflammation, they're suppressing antiviral immune response. And so this was great in a way because it really prepares the body for injury and infection, but it can promote chronic illness. And so what was really interesting about this is that we can see the effects of social factors and adversity both at the epigenome level and at the gene expression level. And they seem to be similar across a variety of exposures. So the work that I'm doing, I'm going to talk about today is in the grant that Ryan mentioned. So this approach to health disparities and cardiovascular risk factors. And I'm going to talk about 3 different studies, all within one data set, which is MESA. And so in the first study, I was looking at examining the influence of socioeconomic status and neighborhood on methylation at genes that are known to be in the stress reactivity and inflammation pathways. And the MESA data set, we had about 1,200 people with DNA methylation. They were about half non-Hispanic white, a third Hispanic, almost a third African-American from 4 different field centers. This is in a population-based longitudinal cohort study that are free of clinical cardiovascular disease at baseline. And there are now more than 6 exams, but we were looking at this data when there were 6 exams. The baseline was in the year 2000. So here we're defining socioeconomic measures as low childhood SES based on maternal education less than high school degree, and low adult SES based on their own educational attainment less than a college degree. So we had 2 markers of SES, and we had 2 markers of neighborhood, and we looked at over the first 5 time points, so the baseline exam and exams 2 through 5, we averaged socioeconomic disadvantage. So this was based on the census. It referred to metrics of education, occupation, income and wealth, poverty. So it was sort of this neighborhood socioeconomic piece. And then neighborhood social environment, which was aggregate level responses within a 1-mile radius on how people felt about their neighborhood in terms of their aesthetic quality, safety, and social cohesion. And we looked at DNA methylation and gene expression in purified monocytes, which is a type of white blood cells, collected at the 5th exam. And so we used, for those of you who know these techniques for methylation, we used the Illumina 450K, which measured 450,000 methylation sites across the genome. And we used a microarray also for gene expression, and it measured about 50K transcripts. And so in this study, we were really interested in, like I said, genes that we already knew had some evidence of, first of all, being in stress and inflammation pathways. And second of all, we looked for genes that had already been shown at that time in previous human or animal studies to have changes in methylation due to these social and psychosocial stressors. So this was the first time that it was really looked at in a population-based study. So I just wanted to point out here that we had these different pathways that we looked at, and that each of these genes had differing numbers of CpG sites, and we specially considered ones that were in the promoter region and in the shore and shelf of those CpG islands that I talked about earlier, because we hypothesized that sites in the promoter region might behave differently than sites outside the promoter region, and the same thing for the shore shelf sites. So instead of testing each methylation site separately, which would have given us this big multiple testing burden, we actually used multi-level models, and we treated the methylation measurements as repeated measures within an individual. And so we modeled sort of the vector of methylation sites as a function of the SES, or neighborhood factor. And we allowed the effects of the SES, or social factor, to vary across the different types of methylation sites. So that was the indicator variables here for the promoter region and the shore shelf region, and their interactions with the social factor. And so we did one model for each gene that we were looking at. And then I'm not going to show this part, but once we found things that were significant, we sort of looked at whether we saw the effects, just in the promoter region, just in the shore shelves, or all across the gene, and things like that, to try to get a little bit more at the mechanism sort of biologically. And so in those two papers that we released, the overall sort of summary of our results are shown here. And so first, in this first column, we have the genes of interest, both in the stress reactivity pathway up at the top and the inflammation pathway at the bottom. And we have the 4 different measures that we looked at across the top. And what I did was, if there was any association between the gene and the social factor and the methylation in the gene, I wrote here, I colored it in purple, and then I wrote the direction of the effect and what type of site it was in. So promoter, non-promoter, shore shelf, non-shore shelf. But the details aren't really important. I think what I really want you to notice is that childhood socioeconomic status seemed to influence methylation of genes in the stress reactivity pathway. And adult SES really tended to be more concentrated in influencing methylation in the inflammation pathway. And then we had neighborhood social environment, which seemed to have effects on both the stress reactivity pathway methylation as well as the inflammation pathway methylation. And in general, we saw that worse neighborhood environment was mostly associated with increased methylation across most of these sites in most of the genes. And we followed that up by, when we found genes that had these significant effects of the social factors on methylation, we also looked at the expression of those same genes. And we found that had neighborhood methylation associations, where the methylation was also associated with at least one transcript of the gene. So it seemed to have sort of these functional effects, right? And so here's just an example where we had this gene, CD1D. It was an inflammation pathway. So social environment seemed to be associated with increased methylation in the promoter region, which was associated with decreased gene expression. And we found in this gene that the gene encodes a protein that presents antigens to natural killer cells. And when you restrict that protein in mice, you get the development of obesity and insulin resistance. And so, so this kind of like helped strengthen the hypothesis that some of these inflammation and stress reactivity genes might be also important for cardiometabolic diseases that we were interested in studying as well. So I just wanted to show you that that was the candidate gene work that we did. And then we started to think about epigenome-wide association studies. And if we looked across the entire epigenome, what do we see as the relationships between socioeconomic status or neighborhood and all of the genes in the genome? So there was some work done by colleagues of ours in 2017 where they looked at 11,000 people of European ancestry. They used 27 different cohorts for this meta-analysis. And they found 9 methylation sites across the genome associated with years of educational attainment at a very stringent p-value, because we're looking at epigenome-wide, so we need to correct for a lot of multiple testing. And they found these CPGs were associated even after adjusting for smoking and BMI, which are 2 potential confounding factors, which are both really strongly associated with social factors and with methylation in general. But that study found that they couldn't really rule out the effects of residual smoking, alcohol consumption, or maternal smoking during pregnancy as drivers of those associations. And there was another second smaller education, EWAS, that actually found that same thing. But there's been few studies of neighborhood characteristics and their effect on the epigenome. So we decided to do that. Again, using the same 2 neighborhood variables in MESA. And we adjusted for age, sex, race, childhood and adult SES. So we're adjusting for those own individual SES markers. We also adjusted for genetics and technical variables. And we looked across the whole entire epigenome at about 400,000 sites. We did use, for those of you who are statistically minded, there's a program called BACON that controls for bias and inflation in epigenome-wide association studies. So we used that. And we looked for associations that had, after multiple testing correction, a Q value less than 0.05. And what we found were that we actually found quite a few CPG sites that were pretty strongly associated with neighborhood social environment. So this is the negative log 10 p-value here. So the higher you are on this plot, these are all the CPG sites plotted by chromosome. So the higher the association on this plot, the more strongly associated the CPG site is with neighborhood social environment. And when we looked at those CPGs that we found, there were 284 that were significant at that FDR multiple testing correction level. We actually looked at whether the genes that were near those CPG sites were enriched for certain biological pathways. And we found that they were indeed enriched for pathways of immune response, regulation of the immune system, and inflammation. And so, so we found that they seem to match our biological hypothesis in that way, and that they were also enriched for certain features in the genome. So for example, enhancers are regions of the genome that are involved in the regulation of gene expression, but they're not right next to the gene like promoters are. And so we found that a lot of the CPG sites that we had identified were in enhancer regions, or regions that were highly expressed. So the DNase hypersensitive sites here means that they're in regions of active gene expression. And so what we only do a lot of correlations and association tests when we're looking at methylation and its influence by social factors, but it's nice that we can see that we are finding enrichment for some of the regulatory regions and regulatory functions when we look at these CPG sites that we found. And to dig into that just one more layer, we were interested if the CPG sites that we found were also associated with gene expression, right? Because that's kind of the hypothesis here. So what we did was we looked at all of the CPG sites across the entire genome. We looked to see if they were associated with the expression of any nearby genes. And we found 14,000 pairs of CPG sites with genes that were nearby to them that had a strong association after multiple testing correction. So we basically picked out from the whole entire epigenome the methylation sites that we think influence gene expression the most. And that was 2.5% of the methylation sites across the genome. Of the ones that we identified in our study, we found 54% of them actually were in this group of the 2.5% of CPGs that had functional effects. So that's saying that our CPGs that we identified, a whole lot of them had influenced gene expression, a whole lot more than what we would expect by chance alone. So, so we feel like that lends credibility that they're actually doing something regulatory in the genome. Okay, so of course, some of the issues here are that the MESA study, the data set we used had 4 different sites where we collected participants from. So we wanted to make sure that that was not a potential confounding factor because neighborhood exposures are going to vary by site. We also looked a little bit more in that paper into health behaviors and especially body mass index for the reasons that I told you before. So I'm not going to show those results, but I'm going to hit on it in the next study that I'm going to talk about right now. So this last study, the last piece I want to talk about is high dimensional mediation analysis. So, so we know, again, that there's a relationship between social factors and cardiovascular risk factors here, but how much of it is going through the epigenome? So how much of it is mediated by DNA methylation? So to start, I first wanted to show you the relationship between our social factors and neighborhood factors and the cardiovascular risk factors in MESA. And so adult SES and neighborhood socioeconomic disadvantage are associated with some of the cardiovascular risk factors. For example, there's an association with BMI. Adult SES is associated with BMI, waist circumference, log triglycerides, log HDL cholesterol, and log insulin. And after we adjust for BMI, we still see relationships between adult SES and triglycerides, cholesterol, and insulin. With neighborhood socioeconomic disadvantage, we see a relationship, even after adjusting for BMI, with triglycerides and cholesterol. And so we see some of these relationships with the social factors on cardiovascular risk factors. But I just do want to note that the percent variability explained by these social exposures is pretty low. So it's about 3% or less. It exists, but it's not huge, especially after, adjusting for individual SES as well as smoking. This is just giving you a little bit of a diagram here of how we think about mediation, and especially high dimensional mediation. So we've got our set of exposures. They have some path, they have some relationship with our outcomes of interest. And we're interested in this path here, which is the indirect effect. So the path A, the exposure influences the mediator, and the mediator influences the outcome. So this entire path is what we call the indirect effect. The part that does not go through the mediator is called the direct effect. Oh, sorry, I'm pointing at the wrong screen, I think. Oh no, I think you can see it. Okay. So, so we wanted to estimate how much of the total effect is mediated here by methylation. Okay. And I know we're running out of time, so I'll be brief, but basically mediation methods with multiple mediators were pretty sparse until a couple of years ago. And so there's been sort of this a bloom of different methods that you can actually use now to do epigenome-wide or other kind of omics-wide mediation. So we developed some methods, some other people developed methods. There's frequentist and Bayesian methods. And I put here a review paper if you're interested in looking at comparing some of those methods. So what we did in MESA is we were interested in that A-B path, again, that I showed you before, A times B, that mediating path. And we were looking at, for those social factors that influence the cardiovascular risk factors, could we actually see which CPGs were mediating that effect? And so we used a couple different methods that are going to look at this mediation pathway, estimated by regression coefficients. We'll call them alpha 1 and beta 1. And there's three different ways that alpha 1 times beta 1 could be null or could be zero. It could be that the A path has no effect. It could be the B path has no effect, or it could be that both paths have no effect. And so one method that we used is called JT-comp. It's implemented in R. It takes into account that composite nature of the null hypothesis where any of those three different combinations of pathways could be null. And what we found is that for the relationship between adult SES and BMI, we had 410 potential CPG mediators that explained about 53% of that association, and together total. And for neighborhood SES and HDL, we found 43 mediators that explained about 23% of that variation. And this is in the second one after adjusting for BMI. And when we looked at what genes were related to those CPGs, we found that for the SES BMI association, they were enriched for genes in antigen processing and presentation pathways, so related to immunity. And in the neighborhood HDL association, we found genes related to inflammation and cell death or apoptosis. In a second study where we used a different method, this was a Bayesian method, we were looking at the relationship between adult SES and hemoglobin A1C, which is a biomarker for diabetes. And in this paper, what we found were two genes, CCDCS4 and CCND2, that had high posterior inclusion probability, which is sort of like in Bayesian, it's strong evidence for these sites, these CPG sites mediating this effect. And we found that in this method, all the tested CpG sites accounted for about 10% of the total effect of adult SES on hemoglobin A1C. And we're about to release a couple papers that are looking at this association in MESA and comparing different high dimensional mediation methods, including this method, BAMA. So to sum up, future directions, I think one huge thing is that we need more rigorous testing of modeling assumptions. There could be unmeasured confounders that are making the mediation relationship look different than it actually is. The other major thing with methylation is temporality. So with all these disease processes, we don't necessarily know whether the methylation changes are happening first or if they're happening after the disease process has already started. And so a really important step is looking at longitudinal associations there. We want to make sure we're looking at the similarities and differences across self-identified race, ethnic groups, and genetic ancestry-based groups, because we don't want to miss things that are important for race or ethnic or ancestral health disparities as well. And so there are some other pieces, more validation, more replication, and just really trying to get at these mechanisms by which the social environment really gets under the skin and leads to disease, and really thinking about the molecular physiology and its relationship to the social environment so that we can develop new treatments and interventions, but also motivate people to work on the social factors and create change at the social level. I think people sometimes feel like there's we can't use biological mechanisms to motivate social change, but I think that we can. I think it speaks to really trying to change policies that change those social factors so that the biology can adapt and not have these long-term consequences. So precision medicine, but also precision public health. And I just wanted to thank people at the University of Michigan, Drexel, and Duke, and then the NHLBI and NMCHD for the funding that we have.